everyone, welcome back to the channel Proactive Prudence. So as I told in the previous class, we did anatomy, and today we'll be continuing with the physiology, and the topic for today is blood. So we'll be seeing the blood physiology. Before I go into the blood physiology, I just want to brief out what all will be covered in this blood topic. So in this video, I'll be covering from introduction till the RBC, that is red blood cell, will be covered in this video. So the introduction part will consist of the composition and the function of blood. So what is the composition as well as what is the work of a blood. Then the second topic will be about the plasma proteins that are present in the blood. So these plasma proteins are present inside the blood. That is a composition of a blood, and what are the different types of plasma proteins and the function of the plasma proteins. And thirdly, I'll be covering the red blood cells, the formation of red blood cells, which is erythropoiesis, and the different stages of how the red blood cell got evolved, and we'll also be covering the factors that is affecting the function of it. Now we'll also see the normal count of red blood cells as well as the physiological variation that is seen in a RBC. Okay, so in the upcoming videos we'll be covering about hemoglobin, jaundice, anemia, WBCs, which is white blood cells, platelets, hemostasis, blood group, and RES, spleen and lymph, etc. So the introductory part of blood, or the composition of blood, and the blood acting as a circulatory fluid. Okay, what is a circulatory fluid? So what is a medium that is like flowing inside your body? There is a fluid connective tissue, there is a dense connective tissue, there is a loose connective tissue. So this fluid connective tissue, which runs inside your body as a blood circulatory system, is involved in various functions as well as composition. So firstly, blood is consisting of a protein rich fluid which is called as plasma. So this plasma has certain suspended cellular elements. Cellular in the sense there are cells inside the plasma. So blood is referred to as a protein rich fluid which is called as the plasma, and this plasma consists of a cellular elements like WBCs, which is white blood cells, RBCs, which is red blood cells, and platelets. So the total circulating blood volume inside the body is around 8%, which is around 5600 ml in a average 70 kg man. Okay, so 8% of his body weight is composed of the blood volume. Moving to the next one, which is bone marrow. So the origin of blood cells is from the bone marrow. So what does the adult bone marrow consist of? It consists of the cells of red RBCs, WBCs, and platelets, which are originating from the bone marrow. And in children, the blood cells are produced actively because they are at a very young age and the cells are at a dividing stage. So these blood cells are formed from the bones, from the bone marrow. So by the age of 20, the marrow cavity is longer, and it also it is also inactive by the time they reach 20. So till the age of 20, the body of the person is going to nourish in production of healthy red blood cells and WBCs as well as platelets. So by the end of 20 years, the upper humerus and femur stop production or decrease the production of red blood cells. Okay, so next point which says about active cellular marrow, which is red marrow, and inactive form of marrow, which is called as yellow marrow. It is infiltrated with fat cells. So that is how a bone marrow appears when it is active and when it is inactive. Moving to the next topic, the continuation of bone marrow. So in bone marrow, is usually seen in the largest bones of the body. So largest bones of the body like femur and humerus. So this bone marrow is involved in the differentiation of cells to form RBC, WBCs, and the platelets. So you can see in the image there is a hematopoietic stem cell which gets converted into differentiated into lymphoid and stem cell and myeloid stem cell in the diagram or the photo which have been put. So the normal 75% of the cells that are in the marrow are usually of WBCs, which is white blood cells, and they produce myeloid series, and the other 25% is comprising of the red blood cell. So the majority of the cells that is produced from the bone marrow is WBCs. So you'll be seeing the different WBCs that are formed in granular and agranular cells. So we'll be seeing in the upcoming classes. Next, moving to the hematopoietic stem cells, which are the bone marrow cells which are capable of producing the blood cells. So initially, before they form a blood cell, there are hematopoietic stem cells. So these hematopoietic stem cells differentiate into different forms, forming a progenitor cell. Progenitor is a cell that is going to be committed to form a final product, that is RBC or WBC or the platelets. So these cells differentiate into another form to form a committed stem cell. Okay, so this is how a hematopoietic stem cells gets converted into a final RBC or WBCs. So moving to the next one, there are separate pools of progenitor cells. So once the hematopoietic stem cell divides into myeloid and lymphoid series, that as you can see in the diagram, so these are the diagrams of differentiation. So there is a hematopoietic stem cell at the topmost corner, then comes the progenitor cells, that are going to give the generation of the next form of cells. So the stem cell is a hematopoietic stem cell, moving to the common myeloid progenitor and a common lymphoid progenitor. So the myeloid progenitors consists of a granulocyte, monocyte, eosinophil and a megakaryocyte, erythrocyte progenitors. So you can see there is a formation of monocyte progenitor, eosinobasophil progenitor, a megakaryocyte or a erythrocyte progenitor. So this granulocyte monocyte gets differentiated into myeloblast and monoblasts. The monoblast and myeloblast then gets converted into various precursors of formation of different neutrophils, monocytes, macrophages, eosinophils, basophils, mast cells, erythrocytes, platelets, and we have B lymphocyte, T lymphocyte, plasma cells. So these are all the cells that are present inside the blood plasma. So that is how the stem cell keeps on differentiating to form a final blood tissue or the peripheral blood consisting of various other blood cells. Okay, so bone marrow stem cells are also the source of osteoclasts, Kupffer cells, mast cells, dendrite cells and Langerhans cells. So we'll be in detail describing about what osteoclasts are, Kupffer cells are, mast cells are, dendritic cells are and Langerhans cells are in the next upcoming classes. Now this hematopoietic stem cell are very few in number and they are able to replace the whole bone marrow. So as in how the bone marrow differentiate and produces the blood cells, it starts decreasing also, but this does not decrease completely. It gets replaced. The bone marrow gets replaced each and every time. So how does the bone marrow gets replaced? Because of the cells that are being dividing continuously inside the patient's body. So when the patient is having any problem with the bone marrow, it can be injected into the patient to replace the bone marrow, as in the case of leukemia or the diseases that is related to the production of blood cells or the abnormal production of blood cells, bone marrow transplantation is being performed. So bone marrows can be transplanted to the patient unless and until there is no graft rejection or the rejection by the patient. Now moving to the next point. So hematopoietic stem cells are not are derived from the uncommitted totipotent stem cells that can be stimulated to form any cell in the body. So these hematopoietic stem cells need not be committed to any particular cells. As you can see, the hematopoietic stem cells gets differentiated into different forms of cells, different function performing cells. So that is why it is called a totipotent stem cells, uncommitted totipotent stem cells. Moving to the next point, which says adults have few of these, but they are more readily obtained from the blastocytes of the embryo. So what are blastocytes? These are the phase during which after the fertilization of the male and female gametes, there is a formation, there is a fertilization of those two. That fertilized zygote gets divided, the cell gets divided each and every time by the process of cell division, and as the cells keep forming, there is a stage of blastocysts that is formed. So this blastocysts get converted into an infant, so because of the continuous cell division that is happening. So the blastocysts are obtained in the embryos. That is what they have described about the bone marrow. Moving to the next topic, which is about plasma, that is the constituent or the fluid portion of the blood. So how is a plasma looking? So you can see in the diagram there is a yellow color fluid which consists of a plasma, salts, minerals, water and proteins inside it. So this plasma is a fluid portion of the blood and is composed of a majority of the blood, like there is a 55% of plasma. So what does this plasma do? What is the percentage of plasma inside a human body? So it is around 5% in a human body, weighing around 5%, and around roughly 3500 ml of the plasma volume is present in a 70 kg man. And plasma clots on standing remain fluid only if an anticoagulant is added. What is an anticoagulant? So if you leave the blood for around 2 to 3 minutes, there is a formation of a thick mixture or a clot formation. That is because of the coagulants that are present inside the blood or the blood components. So this plasma does not clot unless there is an anticoagulant. There is a stop of clot formation. It can stay as a fluid. Otherwise, it can form plasma clots. Okay. Moving to the next part of the plasma. So if a whole blood is allowed to clot and the clot is removed, the remaining fluid is called the serum. So as you can observe, whenever you get hurt, after the blood has stopped bleeding, there is a white color fluid or a yellow color fluid that keeps oozing out from the wound. That thing is called a serum. So the remaining fluid that is coming after the clot formation or the plug formation near the wound is a serum. So what does the serum consist of? It consists of the fibrinogens and clot factors 2, 5 and 8, which are involved in the formation of fibers. So these fibers are going to form a mesh-like structure around the wound, stopping the blood completely. Okay, so it can form a permanent plug formation or the secondary hemostasis, which we will be learning in the upcoming videos. So this is the composition of serum, and serum also has a higher serotonin content, which helps in the breakdown of platelets during clotting. So during the clot formation, platelets play an important role in plug formation or the stoppage of the blood temporarily. So the temporary blood formation is helped by the serum's content containing of higher serotonin levels. So that is how a serum helps in blood clot formation. So you can see in the diagram there is a blood plasma, a buffy coat, then you can see a red blood cell. So there is a serum and a clot formation. So we'll learn about what buffy coat in the next class. Moving to the plasma proteins. So as I told in the beginning of the plasma topic, plasma consists of water molecules. There are proteins inside it. There are salt and minerals also present in the plasma. So when you remove the plasma or centrifugate the blood and obtain plasma, the plasma looks like a yellow color fluid, as you can observe in the diagram. So the plasma proteins that are present inside the plasma are albumin, globulin, fibrinogen fractions. So these are the three different plasma protein fractions that are produced, obtained in the plasma. So this the proteins that are present have are impermeable to the capillary wall. So the most capillary walls are relatively impermeable to proteins in the plasma, and the proteins therefore exert an osmotic force of about 25 mmHg across the capillary wall that pulls the water into the blood. So how is the filtration happening, or the blood is pulling the water or carrying the water that you drink into the kidneys? So this is how it happens. So this proteins are impermeable to the capillary walls. Impermeable in the sense they do not get dissolved with the capillary walls. So when they don't get dissolved, or when they don't react with the capillary walls, they start absorbing the water at a 25 mmHg pressure, osmotic force. So they pull the water into the capillaries, thereby pulling out any excess water present inside the tissue or a cell. Okay, so the plasma proteins are involved as they act as a 15% buffering capacity. They have a 15% buffering capacity. So what is a buffering? It balances the acid and basic nature of the blood. So they stop the acidic nature of blood by acting as a buffer system. Okay, so blood has a weak ionization of consisting of COOH and NH2, which is acidic nature. So to stop the blood acidity, the acidic nature of blood, these plasma proteins are vital to form a buffering system. Then comes the normal plasma pH is around 7.4, which is near to basic nature. Okay, so next is RBCs. As I told, today's topic ends with RBC. So red blood cells carry a hemoglobin through the circulation. So these RBCs are also called as erythrocytes, and they act as a main circulatory medium which is involved in exchange of oxygen and carbon dioxide among the tissues of the body with the lung alveoli. So that is how the RBC plays an important role. So how does an RBC look like, or the morphology of RBC? It is a biconcave disc, and it is manufactured in the bone marrow, or it is originating from the bone marrow. So in mammals there is no nuclei in the RBCs. So they lose their nuclei in order to help in accommodation of the 4 molecules of oxygen. So that is one function or the physiological variation that is from other organisms that mammals have. So these mammals don't have a nuclei containing RBC. So there is no nucleus in the RBC, that you have to keep in mind. So in humans, the RBCs survive around average of 120 days. So the RBC can circulate in your body for around 120 days, and after 120 days it's going to die in the spleen. So spleen is going to take up all the dead RBCs. So the average normal RBC count is 5.4 million per microliter in men and 4.8 million per microliter in women. So that is the count of RBCs. So the number of RBCs that is being said, or that is being calculated, is called hematocrit, or the percentage of the blood that is there in a RBC that is there in a blood drop, that is called as hematocrit. Now each human RBC is around 7.5 micrometer in diameter and 2 micrometer thick, and each contains approximately 29 picogram of hemoglobin. So a human RBC is made up of 7.5 microgram of diameter. So that is the diameter of the human RBC, and it is 2 microgram thick, and it consists of 29 picograms of hemoglobin. So these numbers are very much important when there is a normal count of RBCs are usually being asked in the exam. So normal counts and the abnormal count because of the any diseases that is acquired in the patient is important to remember. So and then moving to the next point, there are 3 into 1013 RBCs per 900 gram of about hemoglobin in a human body. So it is actually 3 into 10 to the power 13 RBCs per 900 grams of hemoglobin. So there is 900 gram of hemoglobin in RBCs that is circulating in an adult human body. Next, moving to the formation of RBC. So how is an RBC formed? So the process of formation of RBC is called as erythropoiesis, which is production of mature erythrocytes from the peripheral blood, which takes place in the bone marrow, and it evolves from the hematopoietic stem cells. So RBC production or the red cell production is influenced or triggered by the growth factors and hormones that are produced by the pituitary gland as well as the gonads. Then erythropoietin. So erythropoietin is the hormone that is regulating in production of the RBCs. So what is the erythropoietic activity that is regulated by the erythropoietin is when there is a anoxia is breathlessness or the low oxygen content in the body. There is a production of erythropoietin, an enzyme which triggers the production of RBCs. So the principle of erythropoietin is that it is being produced by the kidney and they are involved in the formation of RBCs. So the level of erythropoietin is lower in chronic renal disease, that is people who have completely lost the function of their kidney. So the erythropoietin levels are low in those people, as well as people with renal cell carcinoma or the cancer in the kidneys. So in these two cases there is levels that are being increased or lowered based upon the production of erythropoietin. So erythropoietin, what is its role? It goes and acts on the marrow, and the marrow is responsible for production of RBCs by triggering the hematopoietic stem cells to increase the production of RBC. So how is erythropoietin detected in the plasma or the serum? It is done by a ELISA technique, which is immunoassay technique that is used to indicate the production of erythropoietin in the plasma or the serum of a patient. So there is another method which is immunoassay, which is there is use of radioactivity to determine the presence of erythropoietin. So the normal value of erythropoietin that is present in a human body is 10 to 25 U per liter, that is units per liter. So this is the process of erythropoietin. Now coming to the stages of differentiation. So the erythroid series are produced by the hematopoietic stem cells, followed by a myeloid series. So these myeloid series are finally triggered into the progenitor cell, which is a proerythroblast. So this proerythroblast then converts into basophilic erythroblast, then polychromatic erythroblast, then comes acidophile erythroblast, and the final reticulocyte or the next differentiation, which is erythrocyte. So that is the series of or the stages of differentiation of the erythroid series. So the first stage is proerythroblast. It is an earliest recognizable cell in the marrow, and it is proerythroblast or pronormoblast. It is called as proerythroblast or pronormoblast, and it is usually around 15 to 20 micrometer, and it is the largest cell in the differentiation process compared to the RBCs, and they have deeply basophilic cytoplasm. Baso is basic in nature, and they have large central nuclei. You can observe a dark central nuclei inside, and there is a deep blue color of the cytoplasm, which represents the high content of RNA that is present, as well as the active protein synthesis is happening. So the cell is in active state, and it is having a central big nuclei, and it also has a deeply basophilic cytoplasm. Now this cell on maturation loses the nuclei, and it forms smaller and denser, so smaller as well as little heavier, and the cytoplasm, which is blue in color, gets replaced with a pink stained hemoglobin as it differentiates. So this proerythroblast will undergo 4 to 5 replications and forms around 1632 mature RBCs. Now moving to the next stage, which is basophilic or the early erythroblast. As you can see in the diagram, it is a round cell having a diameter around 12 to 16 micrometer with large nuclei, and there is slightly condensed with the nuclei is slightly condensed compared to the proerythroblast, the previous stage of series of erythroblast. Now it also contains a basophilic cytoplasm, and it undergoes a rapid proliferation or the rapid conversion or the division. Moving to the polychromatic erythroblast, which is the third stage of the maturation of RBC. So it is 12 to 14 micrometer, and the nucleus is at coarse and is deeply basophilic. The cytoplasm is polychromatic, and it has a mixture of basophilic RNA as well as acidophilic hemoglobin, and this stage is also at the stage of proliferation. Proliferation is conversion or getting matured. Moving to the orthochromatic or the late erythroblast. This is the final stage of maturation of a nucleated RBC cell, and hence it is called as orthochromatic or late erythroblast. Now this cell has a smaller compared to the proerythroblast and the previous stages or the series of RBCs. So this is around 8 to 12 micrometer in diameter and consists of a small and a pycnotic cell. Pycnotic is a nucleus which does not contain RNA. So which is not consisting of RNA is called as pycnotic nucleus, which has a dark nuclear chromatin. So you can observe there is a chromatin which is dark in color, and the nucleus is also dark. The cytoplasm is slightly acidophilic, and which is diffusing with the basophilic hue in the presence of large amount of hemoglobin. So there is larger amount of hemoglobin presence. So this basic nature keeps reducing, and the acidic nature, because of the hemoglobin, starts increasing at each and every series. Moving to the next part, which is reticulocyte. One stage before it is completely forming a RBC. So this reticulocyte has no nucleus. So this nucleus is finally extruded, which is thrown out of the cell. So this is the stage where there is budding or the juvenile red cells. That is, the cell is devoid of nuclei, or there is no nuclei. It contains ribosomal RNA and is still capable of synthesizing hemoglobin, but is not completely covered with a nucleus or completely dependent on the nuclei. Now this reticulocyte is still in the marrow. So all this process till the reticulocyte is taking place inside the marrow, and after that 1 to 2 days the reticulocytes are released into the circulatory system, and in the circulatory system they get converted or matured into an RBC, which is biconcave shaped. Now this, how is the proerythroblast to the orthochromatic cell dividing? It is completely converting from a basophilic stage to an acidophilic stage because of the presence or the larger amounts of hemoglobin production. There is a nucleus that is being thrown out of the cell, not completely, but yeah, slowly, and the RNA that is present, or the ribosomal RNA that is present, is still allowing the synthesis of hemoglobin. And as and how it reaches the state of reticulocyte, there is around 1 to 2 days that is they spend 1 to 2 days inside the marrow itself, and then they are released into circulation. When in circulation, they go into the peripheral blood, and from peripheral blood they go to the spleen, wherein they are going to mature into a biconcave RBCs. And once they are biconcave RBCs, they come into the proper circulation and functioning of the RBCs. So this reticulocyte is in the peripheral blood cells and are distinguished from mature red blood cells by having a slight or a very few amount of basophilic hue or the basophilic color inside the cytoplasm, which is similar to orthochromatic erythroblast. So the reticulocyte can be counted in laboratory by vitally staining with the dyes like methylene blue or brilliant chrysal blue. So the microscopic images that you are observing over here have the dyes that are stained. So without dyes, the observing of microscopic structure is very difficult. So hence the staining is done with the methylene blue or the brilliant chrysal blue. So this reticulocyte is stained by different methods because they are containing a deep blue reticulofilamentous material. So when they have a deep blue reticulofilamentous material, the staining becomes a tedious job. Then, while erythroblasts are not normally present in human peripheral blood, reticulocytes are found normally in the peripheral blood. So it is not exactly when there is no erythroblast that is present in human peripheral blood, reticulocytes are found normally in the peripheral blood. So while they are at the stage of erythroblasts, they are not present in a human peripheral blood. So these reticulocytes are usually found in the peripheral blood, in the sense they are present at the peripheries of your blood circulatory system, that is near the fingers or the legs. So the erythroblasts are mainly comprised in the central blood circulatory system. That is the meaning of the sentence. Now moving to the normal range of the reticulocyte that is present in adults are 0.5 to 2.5% and 2.6% in the infants, 2 to 6% in the infants. And the percentage of the erythropoietic activity that is happening in these are the percentage of how much the erythropoietic activity is happening in the periphery of the blood, that is the periphery of the blood circulatory system. Now their proportion is increased in conditions of a rapid red cell regeneration, or the proportion of reticulocyte or the number of reticulocytes increase when there is hemorrhage, that is blood bleeding, or hemolysis when there is destruction of the RBCs, or when there is a hematopoietic response due to anemia treatment, when there is anemia treatment. So now what are the factors that are affecting erythropoiesis, or the cause of lowering or increasing of erythropoiesis, that is increased production of erythropoietin or decreased production of erythropoietin occurs due to chronic renal disease as well as the renal cell carcinoma, as I specified in the before. And there is increased production of erythropoietin when there are different types of anemias. So there are anemias of chronic diseases which leads to enhancement or increased production of erythropoietin. So it could be because of the anemia could be chronic in case of infection and neoplastic condition. There is an infection as well as there is a cancerous or the growing cause of anemia. So there is increased production of erythropoietin. Now moving to the polycythemia rubra vera. So this is a condition where there is a erythrocytosis, but there is a depressed production of erythropoietin. Though there is lot of erythrocytes that are produced, the erythropoietin level is at a very low levels, but the amount of production of RBCs is more. That is a condition of polycythemia rubra vera. So there is an abnormality in the HSC, which is hematopoietic stem cell. So there is a problem in the hematopoietic stem cell which is present in the bone marrow, and that is uncontrollably producing the number of RBCs. So this hematopoietic stem cells are not under the direct control of the erythropoietin activity, but there is an abnormality in the bone marrow itself that is producing RBCs and leading to an abnormal number of RBC production, leading to a more number of RBCs in the blood. So other than erythropoietin, androgens and thyrosin can also be involved in RBC production. So androgens are the ones that are produced by the, so androgens are what? These are produced from the hormones, and thyroxin is produced from the thyroid gland. So these are the two hormones that are involved in red blood cell production as well. So coming to the function of RBC, so the function of RBC is to carry oxygen from lungs to the tissues and then transport carbon dioxide to the lungs. So they obtain carbon dioxide from the tissues, carry that carbon dioxide containing RBC to the lungs. So the lungs that have oxygen in them, that are carrying oxygen, get it exchanged with the carbon dioxide that is present inside the blood, and the oxygenated RBC is sent back to the tissues to deliver the oxygen for generation of ATP. So that is a Embden-Meyerhof pathway which you have learnt in your 11th and 12th classes. So this pathway will reduce the power of NADH and NADPH by the hexose monophosphate shunt. So that is a pathway that describes about the function of RBC. Now the oxygen carrying capacity is one of the function of RBCs. So in normal adult hemoglobin, HbA is an extremely efficient oxygen carrier. So HbA is a hemoglobin that is carrying oxygen for with 4 molecules of oxygen with it. So this hemoglobin is a tetramer, so it can carry around 4 oxygens along with it, and the oxygen dissociation curve, or the oxygen, that is how the oxygen is being carried by the cell RBC, and how it is living or dissociating or cutting its bond formation with the oxygen is dependent upon the dissociation curve. So it is represented graphically how the RBC is involved in the dissociation of the oxygen. So that is in sigmoid in shape. So the sigmoid shape is represented for the oxygen carrying capacity. Now moving to the next point, which is oxygen affinity with the hemoglobin is expressed as P50, or the efficiency at which the hemoglobin can hold on to the oxygen is around P50. That is, the oxygen tension which it has with the hemoglobin is around 50%, and the saturation point of oxygen with the hemoglobin is around 50%. Now coming to the pulmonary capillaries which have high PO2, that is, pulmonary capillaries are the capillaries that are present close to the lungs. So these are the capillaries that are present near the lungs. So these have higher PO2, that is, they have higher oxygen tension, because when you breathe in oxygen, there is more of oxygen that is going to the lungs, and these lungs are connected with the pulmonary capillaries, and these pulmonary capillaries in turn help in the exchange of carbon dioxide with oxygen. So these tissue pulmonary capillaries will have lower PO2 because they require oxygen. Hence tissue capillaries have lower PO2 and pulmonary capillaries have higher PO2. Now coming to the extent to which how the oxygen is released from the hemoglobin is dependent upon the 3 factors, that is the nature of hemoglobin chain, the pH, and the concentration of 2,3 BPG, that is biphosphoglycerate. So how is the oxygen that is being carried from the lungs to the tissues dissociating. It is dependent upon these three factors, which is the nature of globin chain, that is how the bond formation between the hemoglobin molecule as well as oxygen is strong or how weak it is. And it is also dependent upon pH. pH is the state or the what is this acidic or basic nature of the tissue is also important in order to exchange or diffuse the oxygen into the tissues. And the importance of 2,3 bpg is also important. The concentration of 2,3 bpg is important in order to release the oxygen from hemoglobin to the tissue. So the first point under these three factors are the normal adult hemoglobin. How is normal adult hemoglobin? It has a lower affinity for oxygen than a fetal hemoglobin. Now comparing an adult and a fetus about the ratio at which they are involved in the attraction towards the oxygen molecule, how the hemoglobin is attracted toward the oxygen. So the fetal hemoglobin has higher affinity than compared to normal adult hemoglobin. So fetal hemoglobin captures oxygen faster when compared to an adult hemoglobin. So they are at a greater amount bound to PO2 of a tissue capillaries. Moving to the next point, the second point, that the pH level. So if the pH is acidic or there is an acidic pH in the hemoglobin with oxygen affinity will be lower. So there is a chances of lower binding of oxygen with hemoglobin when there is acidic pH. So this is called as Bohr's effect. So you have to remember the Bohr's effect, which is very much important in the upcoming classes. I'll be telling about this in the respiratory system physiology. Thereby there is a cause of release of enhanced release of oxygen from erythrocyte at lower pH in the tissue capillaries. So because of the Bohr's effect, there is a enhanced or increased release of oxygen from the RBCs into the tissue capillaries. So it is when there is acidic pH, there is release of O2. That is what you have to keep in mind. Now moving to the third point, a rise in red cell concentration of 2,3 bpg, an intermediate product of Embden-Meyerhof pathway, as occurs in anemia and hypoxia, causes a decreased affinity of HbA or oxygen. Thus in turn results in enhanced supply of oxygen to the tissue. So because when there is a rise of RBC concentration in the 2,3 bpg, in case of anemia and hypoxia, there is a decreased affinity of HbA for oxygen. So when there is a decreased affinity, when there is a decreased attraction of HB or hemoglobin to the oxygen, there is increased chances of releasing of oxygen into the tissues very fast. So that is why 2,3 bpg concentration is also important. So how is the CO2 transported? So CO2 from the tissues is being collected by RBCs into RBCs sometimes by other pathways, which we will be seeing. So the important function of RBC in case of CO2 transportation is from the tissue capillaries, the PCO2 is high. So the CO2 usually enters erythrocyte. Very much of it is converted into bicarbonate ions, which diffuse back into the plasma. In pulmonary capillaries, see, in the case of when the tissue capillaries have these PCO2 in them, there are chances it enters erythrocyte and sometimes it might get dissolved inside the blood itself, the plasma itself, forming a bicarbonate ion, which sometimes does not get into the RBCs because of the higher concentration of H plus in the blood. So they end up retained in the plasma. So the amount that is going back into lungs or the pulmonary capillaries is reversed. The process is reversed and the bicarbonate ions are converted back into CO2. So once the pulmonary capillaries reach the lungs, the bicarbonate ion inside the plasma, not RBC, inside the plasma gets reversed back into CO2. So there is a chemical reaction that is causing the CO2 formation from HCO3. So when there is H2CO3, when the H2CO2 is converted into CO2, leading to formation of CO2 back, and that CO2 is collected by the lungs and sent out in the form of exhaling or expiration. So this CO2 that is produced by the tissues that this CO2 that binds with hemoglobin is called deoxyhemoglobin. That is deoxy, that is there is no oxygen. Deoxy is referring to the CO2 combining with the hemoglobin. It leads to formation of carbaminohemoglobin, or the carbon that is combining with hemoglobin, forming a carbaminohemoglobin. So the normal count of RBC is 5.5 plus or minus 1.0 into 10 to the power 12 per liter in men, and 4.8 plus or minus 1.0 into 10 to the power 12 per liter in women. So the packed cell volume, or the hematocrit volume of erythropoietic erythrocytes per liter of a whole blood is around 0.47 plus or minus 0.07 liter in men, and 0.45 plus or minus 0.05 liter in women. So that is the packed cell volume of PCV, or hematocrit, the volume of erythrocyte in a liter, in per liter of whole blood. So you're calculating by PCV method. The other one is the hemoglobin content, or the amount of hemoglobin that is present in a normal human body. In men it is 15.5 plus or minus 2.5 gram per deciliter, and in female it is 14.0 plus or minus 2.5 gram per deciliter. So the range is from 11.5 to 16.5 gram per deciliter in women, and 13 to 18 gram per deciliter in men. Based on these normal values, you can determine whether the patient is suffering any problems or any diseases like anemia, or there is a cancer, or there is a problem with the bone marrow. So these are the normal values that determine the abnormality in the patient's blood system. So the methods that are used to calculate the mean corpuscular volume, or the volume of corpuscles or the cells of RBC, is PCV in per liter by PCV is packed cell volume by the RBC count in a liter of a blood. Then the normal value of MCV, that is mean corpuscular volume, is 85 plus or minus 8 femtoliter, or 77 to 93 range of femtoliter. Now the second method of measuring a mean corpuscular hemoglobin, or the hematocrit, or the hemoglobin levels in the corpuscle or the cell, is HB per liter, that is hemoglobin per liter that is present inside the blood, by the RBC count, the number of RBCs in a liter of blood. Now the normal range of MCH is around 29.5 plus or minus 2.5 picogram. So the levels or the range of MCH is 27 to 32 picogram. Moving to the mean corpuscular hemoglobin concentration, or the concentration of hemoglobin in the cell, as measured by HB into PCV, that is the amount of HB into the packed cell volume in a liter by the deciliter of deciliters of blood that is present. Okay. The normal value of mean corpuscular hemoglobin concentration is 32.5 plus or minus 2.5 gram per deciliter. So the range of MCHC is 30 to 35 gram per deciliter. So this MCHC is independent of how the red blood cell count is, or the size of it is it is not dependent on it. So the MCHC is involved in the values or the indication of how low the iron is in the anemic patients. So it is usually used to diagnose whether there is a macrocytic anemia. So this is important. So all these three values, that is the mean corpuscular volume, the mean corpuscular hemoglobin, the mean corpuscular hemoglobin concentration values, normal values, are very much important. They have been asked in exams many times. So concentrate about the values, normal values of RBC or the hemoglobin values. So these three are important. And moving to the last method of measurement or counting of RBC, which is red cell distribution width, RDW, is in assessment that is used to determine the volume of our red cell or the size of the red based upon the size of the red cell. For example, if a fragmented red cell has a tiny size like macrocytes and reticulocytes, which are large size. So in the cases of RBCs or red blood cells that have been forming tiny pieces from the macrocyte or the bigger RBC that form smaller pieces because of these bigger sizes, it is determined by red cell distribution width. So you're technically using the size of the RBCs to determine whether there is a problem with the red cell size. Okay. So the physiological variation that is happening in RBCs are when the person lives in a higher altitude, or a person who lives in a mountains on the mountains, they have a greater number of RBC production because there is lower oxygen production, oxygen present in the air in the higher altitudes. So hence these people will have higher number of RBCs, which these higher number of RBCs are produced in relation to the lower oxygen in that atmosphere. Into the next point, which is tissue oxygenation is the most essential regulator of RBC production. So the level of how the oxygen is combining with tissue to release energy is dependent upon how the RBC is being produced. So this tissue oxygenation determines the amount of RBC production. Now moving to the last point, which is erythropoietin stimulates the red cell production, and the formation increases in the response to hypoxia. Now, how is this RBC production stimulated? By erythropoietin or the enzyme erythropoietin. Now, when is this erythropoietin going to increase in the amount? When there is hypoxia. What is an hypoxia? The case where the patient is not receiving a normal amounts of oxygen for breathing. That is a case of hypoxia. When in response to hypoxia there is a trigger of erythropoietin, in response to erythropoietin there is production of RBCs. So the upcoming topics will be covered in the next class, that is the hemoglobin will be covered in the next video.